Next one. Okay, so the biggest thing here is that normally with an insect and a plant, it's quite easy to control something because you just spray the insect and you're done. The biggest issue that we have is that Liberibacter bacterium. So that it can't go anywhere on its own. It needs the psyllid to go somewhere, and but it can transmit it so quickly that you have to be so fast, with managing it that it's almost impossible to not get any zebra chip in your crop. Next slide, please. So in New Zealand, when we tested in 2012 or something, we tested the haplotype. So you've got Candidatus Liberibacter solanacearum. It's a long word, 3 words. And there are some versions of it, and they're genetically slightly different. So there's a lot of letters, and it's a table later on. So we've got haplotype A, which is also found in the US. The bacterium can live in the phloem of the plant, and it can live in the psyllid. In the phloem of the plant, that's what it uses to get around, basically. And in the insect, it goes around as well. And there's a little insect there, and you can see where people have actually looked at where the Liberibacter goes in the body. The most important part is that once it is ingested, it goes through its body, and then it ends up in the salivary glands. Once it is in the salivary glands, it just stays there and it replicates. So once the psyllid is positive, it will basically not get negative anymore. So it stays that way. Nymphs can become positive by feeding, but adults can become positive by feeding as well. The biggest issue that we have is that you can't grow Liberibacter in a petri dish like we do with some fungi. So everything we do, we need a molecular test to see whether the bacterium is present. And I mean, in the spuds and in the psyllids themselves, in plant material. The interesting part is, and this is quite similar to what we see with aphids as well, is that when you take a population and you take like 100 psyllids from the field, you find about 3 to 5% of them positive for Liberibacter. That's the same what we see with aphids and viruses. So it's actually a really low percentage of the population that is negative. However, if you have volunteers out there, for example, and those are positive, the psyllids will be attracted to that plant. Once you start testing the psyllids on that plant, of course, you find a much higher percentage of psyllids positive for the Liberibacter. So those volunteers are really important, to actually manage, because that it keeps that Liberibacter in the environment. What the Liberibacter does as well is that it changes the behavior of the psyllid. And we have shown that if a psyllid is infected with Liberibacter, it's actually more attracted to clean plants than to Liberibacter infected plants. If you are a psyllid that is not infected with Liberibacter, then you're more attracted to Liberibacter infected plants. So it's also manipulating the plant and the psyllid to make sure that it gets around in the environment. What it also does is it makes the positive TPP, so the ones that have Liberibacter, they start to drool a bit more when they feed, and that means that they, in the drool, basically, when they're feeding, it means that they put all the Liberibacter in the plant when they do that. So that's all, something that the bacterium does to the insect. Next slide, please. So, like I said, there's many Liberibacter pathogens. So you may have heard of the citrus greening disease, Halongbing. That is Candidatus Liberibacter asiaticus or africanus or americana. There's also Candidatus Liberibacter europaeus, Brunwickiensis in Australia. So there's quite a few of them. The most interesting part is that when we look at our one that attacks potatoes, it has a lot of haplotypes. And they give them letters to indicate the haplotypes. And haplotype means that they're just slightly genetically different from each other. So they still have the same name, and then we say haplotype A or B or F. So what you can see here is that, you know, we have quite a few different ones found now. And not all of them attack potato. So we've got A and B, and recently there was F as well that can live in potato. F, they haven't seen any symptoms yet, but A and B, we know that they produce symptoms, right? We got A here that produces seborrheic disease. B is a lot more severe apparently in the United States, so it has more of an effect on the plants. And yeah, these are just all the different plants, and most of the ones that are not A or B have a different insect vector as well. They're all psyllids, but they're just different kinds of psyllids. We have in New Zealand detected the CLSO, so our Liberibacter, in quite a few different host plants, but also some weeds. So that means that out in the environment, we can have those sources of Liberibacter. And the interesting thing is, because this seems like a fairly recent kind of thing, right? When was it, 2009, that we found out that this new to science pathogen existed? Well, we had, after we've done a lot of work more recently, there were some people in the UK and they tested some really old seed lots from the early 1970s. And they found CLSO in there. They found basically the potato Candidatus Liberibacter in there. So that means that this pathogen has been around for a long time, and something must have changed in our varieties or in our climate or some or with the vectors, that all of a sudden it becomes such a problem for us. Next one, please. So what happens in the plant? I'll just gonna reduce this to a very short statement, and just it manipulates the plant. The plant responds to the psyllid, but also to the Liberibacter, because it feels like it's being attacked. There's something foreign in their body, just like we do, you know, when we make antibodies. The plant responds, and it has certain pathways to do that. And as a result, you start seeing all those funny, you know, leaf curling and cupping and aerial tubers being grown. So that's really what's going on. Also, what the plant tries to do, it knows that there's something in its phloem that's trying to travel around. And what it does is it's trying to close the phloem. So the phloem is basically all cells that are stacked onto each other. And so there is a little door between those cells, and the plant just closes those doors. So as a result, you know, your plant's not feeling too happy, basically. Next one, please. Oh yeah, and the thing is, if a psyllid feeds on it, it takes about 3 or 4 weeks to see that plant response. So once you see the symptoms, you know that 3 or 4 weeks ago you had positive psyllids in your crop. So the psyllid gets positive by feeding on a plant that has Liberibacter in it. Like I said, it can be the nymph feeding, but it can also be adult feeding. And you can totally understand how that works in the field. There is a positive psyllid, it lands on your potato or tomato or tamarillo. It starts feeding, it makes the plant positive. In the meantime, the female may lay some eggs, those emerge, the nymphs start feeding. And what do they encounter in the phloem? basically the Liberibacter that the mother put into that. So very quickly you can see how that whole family gets infected with Liberibacter, and once they become adults, they will disperse. When I said that it had to end up in the salivary glands of the psyllid, it takes about 2 weeks to get there, so there is a bit of time before it can start transmitting the Liberibacter. Let me see. Once it can transmit it, it's 5 minutes, and then it has put the Liberibacter in the plant. Yeah, thank you. Next slide, please. So the psyllid, there's a primary infection, and then with potato seed, it's secondary infection. But it's at a very low level. And can you just flick slowly through those boxes, Francis? So there was some work in the US where they said 0.3% of positive tubers that are positive when they grow from a positive mother tuber. Andy Pittman did some work here in New Zealand. So they planted 62 positive tubers. They ended up with 56 asymptomatic plants. 2 developed foliar symptoms. So they're those small gnarly plants that you see with lots of symptoms. And 4 tubers decided not to sprout. Next one. And then at harvest, they could detect from those 56 asymptomatic plants, they could detect LSO in 39 of them. They found the LSO in 5 daughter tubers, and then 1 tuber actually had the zebra chip symptoms. So it's quite an interesting mechanism, but that whole seed transmission is relatively low. Next one, please. These are just the symptoms in the different plants, just depending on what kind of grower you are. So just flick through those because they don't need a lot of explanation, I think. These are the first symptom is the cupping of the leaves. We all know that virus can look like that as well. So in one of the later slides, we say, you can't go into a field and go like, oh yeah, boom, that's Liberibacter. Very often you have to go by deduction and go like, okay, are there aphids around, and check some like the stems and everything as well. So the internodes start to shorten because of Liberibacter, and that's so with the new growth, the internodes get shorter and shorter, and that's why you get a really bushy kind of symptoms when you look on top of the plants. Next one. The swollen nodes and the aerial tubers and the bud proliferation as well. Just a very thick-looking stems and everything. Aerial tubers, we can see that with Rhizoctonia. So again, it's not just a Liberibacter thing. Next one, please. So these are the symptoms in other crops. The people here have been given a handout, but it looks relatively similar. There's lots of cupping. I think Tamarillo looks a bit different to the other crops, basically, but I've never seen an infected Tamarillo tree in person, so I can't comment on that too much. But for tomato and capsicum, absolutely, you get the cupping of the leaves. And for a capsicum, my experience is that it basically stays like that. There is not much more going on, whereas tomato can just basically die because of Liberibacter. Next one, please. This is the bushy growth in Tamarillo, so kind of a similar response as potato. Next one. Like I said, it can lead to plant death, and that's, I think, how they found it here at the Pukeko Research Station in 2008-9, because all of a sudden those plants started to die quite quickly. And you can see that even the Tamarillo tree, they will basically die because of Liberibacter infections. Next one, please. Seed infection. How can you see that a seed potato is infected? Once it starts sprouting, it has those really thin, watery sprouts, and quite a lot of them as well. The tuber feels a bit flaccid. It feels a bit spongy. It will grow stunted plants like the one you see there on the left-hand side. And that happens in the field as well, and they can eventually get overgrown by the healthy plants. One thing, once you start digging up some potatoes halfway through the season or at the end of the season, and you can still find the mother tuber and it's rock hard, then you're pretty sure that that was a positive mother tuber. And like we showed before in one of the slides, that those infected mother tubers can actually grow healthy plants. Next one, please. This is how it spread through New Zealand. So in 6 years' time, that was basically from north to south. So that went really quickly. Next one. So this is more about the psyllid. So we start with the adults. The adults are the only life stage that is mobile. So the nymphs cannot fly. They can walk, but only the adults can fly around in the environment. Next one, please. Adults will lay eggs. They're on little stalks, just like lacewing eggs, but a shorter stalk than a lacewing. And very often you see them on the outside of the leaf as a little halo. Most of those eggs are laid on the new leaves. So when you scout the plant, look at that, the new leaves at the top, and you can see those halos sometimes of eggs. And that's a photo of boxthorn. Can you click through, please? In Southbridge, and there's just gazillions of eggs there. So that wasn't very good. And then those eggs develop into nymphs, and they have 5 nymph stages from very small to large. Another click, please. So those small ones, they're pretty sessile, so they don't really move a lot. Those bigger ones can actually really walk. They're quite funny, and they can be quite fast as well. So they can walk, if plants are overlapping, they can walk from one plant to the next if they want to, or walk within a plant. Next one. Something really important, because most people are familiar with the aphid flights. TPP doesn't have a flight. There is no seasonal flight. There is no flight early spring or whenever that they go from one host to the next host. They're just around in the environment. They fly around. We've caught them in blackcurrant crops. We've caught them in big dairy paddocks. So they're just in the background always. And if they land on a potato and they start feeding on it, they go like, oh, okay, cool. I can stay here. You nicely irrigate your crop, so it will never dry out. So it's nicely fertilized. So why would it move on? Next one, please. You heard Francis talk degree days, and that normally we use that for insect development. So very often people do experiments where they put them at a certain temperature and then see how long it takes to go through the different life stages. So this is our lab colony. It's at 25 degrees. And this is kind of like the number of days that it goes through a generation. And it collects degree days. So everything above 7 degrees Celsius, you start kind of adding that up. And next one, please. So here, this is, I couldn't find any field data from New Zealand. I'm sorry about that. So this is from Texas. And you can see this is out in the field. And you can see that it's quite a different pattern of the life stages. And that is because when the temperature is different, the speed at which the insect will develop is different as well. So when it's cooler, it develops slower. When it's hotter, it develops quicker. That's why that December, January period is quite important, because that's where we see basically the biggest change in temperature all of a sudden. And the psyllid starts sticking over quite a lot quicker. Next one, please. So in New Zealand, we have the West. So again, the psyllid is divvied up in haplotypes, not just like the Liberibacter. And they, in the this is all United States vocabulary. So they've called it the Western haplotype. So that's from the California area. That's very likely where our psyllid came from. One really important thing is that when the adults come out of the nymph, they are very pale. They are kind of yellow-green, and they don't have that stripe. And then they start to harden up, and about 3 days later, they will be that brown color and have the white stripe. The feeding of Liberibacter negative psyllids. So very old and some more recent material from the US say that there is a toxin. They've never found the toxin in the psyllid. They said that the toxin would develop, would induce the plant do not get the same observations here in New Zealand. And one of the reasons might be that because back in the day, the molecular method to look for Liberibacter in a plant was not that sensitive. So the threshold of detecting was much higher. So you needed like maybe 1,000 cells of Liberibacter to detect it, whereas now the test that we have, we can go down to 6 cells of Liberibacter basically. So a lot more sensitive. So maybe there's something there, or maybe there is a difference in the genetics of the different potato cultivars. And then we have native psyllids as well, which is really nice. But there is one that looks like TPP, and it is one that always comes in Canterbury, I must say, it always comes in earlier than TPP itself. Can you click, Francis? So, and you cannot just look and go, oh yeah, that's the other one, because they're so lookalike. The only difference is, and you can see it here, so on the left-hand side is a psyllid with its legs hanging down, and there is something towards the end of the leg, we call them spurs. Can you click again? Where TPP looks slightly different to the Pittosporum psyllid. And you need a microscope for that. You cannot tell them apart. So the people like Fruit Fed who do the assessments of sticky traps and everything, they are well aware that they can't just go by the white stripe on the psyllid. They actually have to look at the legs of the psyllid to make sure that the right one. Lots of unknowns still. I know that you still, after all those years, I know that you will talk about it as well, I think, Roger. Yeah. We don't know how the presence of predators and parasitoids affects the zebra chip disease, well, if there is like a threshold or something like that where you go like, okay, this is how it works. We don't know why there are zebra chip hotspots in certain areas within New Zealand. We don't know how exactly the Liberibacter manipulates the insect and the plant. There is a lot about insect vision and about smelling as well. We got some really good results in the lab, but the biggest thing is that once we go out in the greenhouse or in the field, we lose all of that. So, and that might be because the psyllid uses a different cue, whether when it's flying than when it's walking, for example. So when it's flying, it might use its eyes. When it's walking, it might use a more like its mouth parts to actually go like, hey, is this what I want? So Yeah, work in progress, I think. Next one, please. I think this one must have been hammered home quite well, right? Yeah, flick through it, Francis. Really important, especially for seed crops, because they just keep resprouting, and the psyllid, if there's one little green leaf, it will find it. Next one. This is what we found in one of the crops. It was an organic grower, and they had roped some tubers or plants earlier on in the season, and they put them in a bag next to the paddock, and then it started growing plants again, and they were just choco with psyllids, and very likely Liberibacter as well. So that was an amazing source for the next crop that was going to go in. Next one. Monitoring. Will you talk about that?
Yeah, Francis.
Yeah. Okay. Don't drive past it or run past it or fly over it, because you won't see it. You know, you really have to go into the crop. Next one, please. Because that's when you see the psyllid sugars. You know, the white stuff on the. You see the eggs. You see the adults. You might see the nymphs when you look at it. So. You know, take note of that. Next one. And that was it. Thank you.
I do have a couple of questions.
Yes.
I think the one that's relevant for you, I'll just read it as it is could it be a change in the Liberibacter that changed the virulence and the impact of the problem? So I'm guessing that person's asking if there was like a mutation and that led to it being more virulent.
If there was a mutation, then using the, possibly we would have picked it up when we would do the molecular analysis, but it could be that the mutation is in a completely different area. But I have in New Zealand, I have not heard or seen a change in severity or change in disease progression in potato crops. So I don't know. We haven't tested whether there are any mutations, but we haven't heard of any weird things, basically.
Because was that CSLO in that seed 52 years ago? Obviously, it had some genetic comparison, so you could actually identify it.
Yes. Yeah, I will have to go back to that paper and actually see if they find any genetic differences. Yeah.
Not sure if this is maybe a question for Francis. How far can you push the replacements with oil?
How far can you push them? Can you expand on what you mean by how far?
From online.
Is it a chat?
That would be from alternating insecticide with an oil, right? Can you push that further than? I'm assuming it's going to be. Can you push that further than week on, week off?
Ah, right. Yes, we did actually do that, and Peter can speak to that if he's in the room or if he's ducked out.
I think he's just left.
That's such a good question for Peter, but we did have trial areas where it was more than alternating. What we did find is that there was a reduction in yield, and I would suggest that that's because it has that protectant layer that perhaps is potentially could interfere with photosynthesis, so the leaves are not picking up as much sunshine as they are without. So we did look at that. Alternating was, I mean, we looked at doing it every week, but the yield was definitely impacted by that. Does that answer your question?
Possibly it's online.
I'd be interested to know what your thoughts are around Liberibacter moving on equipment. So if you get fallen on a knife or a tractor and you snap something or you break something, can we move that down the crop or in through the next one?
As far as we know, it's basically dead once it goes outside of that phloem tubes. So as far as we know, it cannot survive outside of the insect or outside of the plant. Yes.
Two
No.
questions. First one is it's either very easy or it's not. Why would the Australian psyllid not have Liberibacter?
I called them lucky bastards yesterday. Twice, really. I was talking to some Australian potato growers yesterday, and you're just lucky. Like I said, there is 3 to 5% of a population is positive for Liberibacter, so there's basically more psyllids without Liberibacter than with. We were the unlucky ones that we got positive psyllids, in New Zealand. For them, it must have been a negative one.
The other question.
Yeah.
If I have a potato plant and a hot psyllid lands and starts feeding, and it takes 5 minutes for the Liberibacter to move from the psyllid to the potato.
Mm-hmm.
At what point is that whole plant now hot? And does that differ if I move from a potato to a tomato to a tamarillo?
Good question. They have, so Rebecca Frampton, I don't know if you remember her, she did some of that stuff, and they infected the plants at different times and then harvested them at different times and looked at Liberibacter at all the different levels within a plant and the different organs. But it takes quite a while for the whole plant to have Liberibacter in it. A couple of weeks, 2 to 4 weeks, I think, is that it takes for a whole plant to be positive for a potato. So I don't know how that works for a tamarillo, for example.
It's still short enough that psyllid laying eggs, those eggs hatching 1, 2, 3, 4, 5 instar of nymphs, they're potentially still going to get hot from that.
Yes.
That infected.
Yep.
I think Joe Muyaneza was saying with desiccating seed crops, right, if you had some regrowth and the time that you're getting skin set was enough on that sort of small green leaflet to go down into the tube, right? So it's sort of 7 to 21 days from top to bottom. Question, Jessica.
Yep.
Can you transmit, obviously you've got the psyllid with Liberibacter flowing through its body. Can it transmit Liberibacter to its eggs?
Good question. So in the literature, they say it can. The problem is that they never separated the mother's feeding from where it lays, where she lays her eggs. So you can see that when she feeds on the leaf first and then she lays eggs, that so the way the egg works is that the little stalk is actually in contact with the plant. There is like some fluid going back and forward between that stalk and the plant. So if you have the female feeding first, or the mother, then she lays eggs, and if there is movement, then we don't know whether that Liberibacter would have gone into the egg, basically. Let's put it that way. So we've tried to, for the psyllid to lay eggs on some plastic. And that was really hard, just to disconnect that feeding from the egg laying. And so I would say we don't know, and that's just because I know that there is communication between the stalk of the egg and the leaf. Yeah.
But it does transmit from nymphs into the adult. So if a nymph is hot,
Yes.
It's probably a silly question, because I'm not a biologist. The inability to culture Liberibacter, is that a function of the organism or a function of our technology to be able to culture?
So quite a few people have tried, and we have tried as well. It needs a very specific diet. And the interesting thing is that there is one Liberibacter, I think from papaya or something, where they said, oh, we can actually culture it. And as a result, so if you can't culture an organism, it gets the candidatus thing in front of it. So they said, oh, we can do it. So, but there has been an outlier. So none of the halong bing or the potato ones have people been able to, culture them. And there's a lot of diets going around where they go like, okay, this is what the Liberibacter needs to grow and to function, but nobody has cracked it so far.
So it's not that
It may not
maybe,
be not possible,
yeah.
but it's just really hard to find the right diet that it needs. Yeah.
In terms of the effect of Liberibacter on the adult itself, is there a fitness effect on the psyllid from carrying the disease?
So the Americans have done research on that, and they say that it reduces the number of eggs that a female will lay in a lifetime. Yeah, if they're positive, yeah, it will reduce the number of eggs. We have a positive colony. We started that, it was basically started in Auckland, so we got some stuff from Frances and her colleagues. And it has been continuously in colony, and we just feed it, new plants every single time. And there's still gazillions of them. So I'm not too sure. Maybe there is some, but we can really easily just, since 2010, basically have that in continuous rearing in the lab without crashing. Yeah.
So it's not going to die out.
No, no. Sorry about that. Question over there.
Yeah, I have a few questions, but the first one is regarding the oil. Do you have any more data on which life stage are you targeting there? It's effective against the 7 of them. It's targeting the adults. Because, for example, when you use oil, plant soap, and older paste, there is an efficacy range. Like, for example, apple white fly, which is a very common one. If you use the plant soap, you're mostly targeting adults. So do you know if the oil is actually doing something on the other life stages, like eggs and insters? Or
I think we looked at that. Can you remember, Frances?
Sorry, can you translate that? It was just coming through a little bit echoey
yes,
for me.
It's from back in the room. So the question is like, do the oils work on the different life stages, or is there only certain life stages?
Oh no, I think it's impactful on all of the life stages. Yep. I mean, it's a barrier effect as well as a treatment effect.
And I think what was started with those oils is that once it covers a nymph, you kind of smother it, so it can't breathe. So that's not really, it's not good for them. Let's put it that way.
Yeah.
Can I just jump in with another oil question, and Frances will come back to you. So this is just going back to perhaps a question that I didn't quite understand. If using just the oil, does it reduce, and it reduces the yield, sorry, but does it also keep Liberibacter low? So
we've only done lab trials with that, no field trials. In the lab trials, we've shown that there is a reduced transmission with certain oils. So there was a lower number of cells of Liberibacter that we could find in those plants. But I don't know whether in the field that would translate into less abrachip.
All I would add to that is that throughout the trials that we ran in Matamata, is that we tested psyllids from all of the traps over that period for 3 years, and there was a reasonable amount of pressure on the crop. It would be difficult to conclude anything from any reduction in Liberibacter, but there was Yeah.
And you
Can't
still
quite
got
answer that.
you still got zebra chip in those trials that year?
Yeah, we did.
Yeah. Just
And the reduced and the standard. So
Yeah.
I'd have to go back and look at the data, but they were very comparable. So be a bold statement to say, but I'd like to have a good look at that.
yeah.
Francis, did you have other questions?
Yeah.
Oh, sorry.
So you mentioned that the bacteria won't survive on the environment. But what happens if you have a crop worker that's going across the crop with, say, a pencil knife and going very quickly? There's going to be some.
So they tested that really early on, and they haven't been able to show that it is mechanically transmitted, because that's what you would call it when you transfer it with a knife or with your tractor or something. So it has a very short shelf life once it's out of the plant or out of the psyllid.
But unlikely.
Yeah.
What I did notice over the years of walking in potato crops into the area where you've got your sticky trap, which might be sitting within your crop, is there's obviously mechanical damage caused by the human walking into the sticky trap, is that sometimes we did see more patches of Liberibacter around where the sticky trap was. So it's almost as if the insect senses a vulnerable, slightly damaged plant, and is a bit of a, I don't know, we would probably say that insects being a bully. Insects are sort of known for attacking weaker plants. So if you're looking at your sort of general management strategies, as if you've got a really healthy crop, you might be less vulnerable to the attack of the insect. But we certainly saw more symptoms around the areas where our traps were regularly. Yeah.
They're really attracted to slightly more yellow plants.
And just damage of plants, I suspect, yeah. So where your tractor rows are going down, for example, they might be, you're creating an edge, obviously, with those tractor rows, and those edges, those plants that are being sort of run over might be creating an invitation to the psyllid.
We're crossing over to some of the stuff that Roger and Lex.
Exactly.
I think
right.
I think we can
we'll ask, and then we can keep questions going after.
absolutely. We'll have another question session at the end. Thank you, Jessica.